Hey chat! In the past few weeks, we've talked about gravimetry and titrimetry, which are the two most fundamental quantitative chemical analyses. This week, let's learn our first type of qualitative analysis, chromatography. Chromatography is a technique of separating substances from each other in a mixture. It's qualitative because we can determine the presence of a particular chemical in our sample if we compare it against a standard. Actually, modern forms of chromatography are also quantitative, but let's talk about them some other time. You might remember an experiment when you were younger where you used paper to separate the pigment in ink. This is called paper chromatography, but it's now mostly used to teach chromatography to students. In laboratories, we have replaced it with thin layer chromatography, which is what I will be talking about today. Instead of paper, TLC uses a sheet of glass or plastic with a thin coating of adsorbent, such as silica. This sheet or plate is called the stationary phase. It does not move, and it should also not be disturbed during the analysis. It is on this plate that we add our sample that we want to separate. We first label a baseline where we place our samples, and then we place our samples as spots using these thin capillary tubes. After we prepare our plate, we submerge it in a solvent mixture. This is a chromatography chamber, which is sealed so that the system won't be disturbed, and inside is a mixture of ethyl acetate, which is slightly polar, and hexane, which is nonpolar. Once we submerge the plate onto the solvent mixture, making sure that the baseline is not submerged, the solvent will slowly creep up the plate. Because solvent moves, we call it the mobile phase. Over time, the solvent mixture passes through the spots and goes beyond, dragging along the sample with it. Let's think about this at a molecular level. Our sample contains numerous chemicals which have their own inherent polarities. Like in basic chemistry, like dissolves like, right? Since the stationary phase, silica, is polar, polar chemicals in the sample will interact strongly with it. Those that are not as strongly held or those that are not as polar would be brought up by the solvent mixture instead. So over time, the different preferences of the mixture components to both the stationary phase and the mobile phase will cause their separation. The interaction of a specific chemical with the stationary phase should be consistent across different samples. Here is a test run I did with black marker on the left and red marker on the right. After chromatography, we use a pencil to trace the line of the solvent, called the solvent front, and we also encircle the spots. We can describe the individual spots using the retention factor, which is the distance traveled by the spot divided by the distance traveled by the solvent. As you can see, the retention factors for the same marker should be equivalent across trials. As you can also see, there are no spots across black and red markers that match. This means that the ingredients are different between these two. There are two more things we can talk about from this plate. First, notice how I did the same sample twice, one with a thin application and another much thicker. We want to make spots as small as possible because the thicker the spot, the messier it becomes because it starts streaking. This could make interpretation very difficult. Second, notice how there seems to be unresolved yellow and red spots to the left. The shorter the stationary phase, the less likely it would be that the spots would separate from each other. This is called having poor resolution. You could try making the chromatography sit for longer, or you could also change up the solvent system. One of the advantages of thin layer chromatography, if these plates were store-bought, is that they are coated with fluorescent dye. Since some organic molecules cannot be seen under visible light, if we subject the plate under ultraviolet light, the spots should cover the fluorescent stain, making the spots visible. Thin layer chromatography is a very quick and very simple way to verify presence of particular chemicals. For example, if you are synthesizing a drug in the laboratory, simply spot a pure standard substance to the left and your product to the right. Existence of two spots with the same retention factor should give you evidence that your synthesis was successful. It must be said, however, that thin layer chromatography, which is planar, is just a simple application of chromatography concepts. We can actually also use machines to analyze more complicated types of samples, even helping us quantify them, which we will discuss in the next lessons. We can also use a column to separate the components of a mixture and actually retrieve them, and we will discuss this when we talk about natural products chemistry in the future. But overall, chromatography is very helpful for chemists to be able to prove the presence of individual chemicals in a complicated mixture. I hope you were able to learn more about chromatography, and I'll see you next time.